This is Lecture 2 in BBSP 710. Today we're going to discuss experimental design, specifically controlled experiments, why controlled experiments are important, and what's the proper way to conduct a controlled experiment. Controlled experiments are considered to be the gold standard of experimental design, and whenever possible you should try to use this design for your own experiments. So to explain why controlled experiments are important, let's contrast them with what's considered to be one of the weakest forms of scientific evidence, which is called anecdotal evidence. Anecdotal evidence is a story or an example of something interesting that happened. You will occasionally even see it in the scientific literature that a physician will write a paper about an interesting patient they had or they tried some treatment that works. And it does have at least a certain amount of value, but it tends to be highly unreliable. An example of how anecdotal evidence is reliable is prayer, or intercessory prayer specifically. Probably most of us have heard stories of someone who was sick, had someone pray for them, and they made some type of miraculous recovery. Does that imply that we should get rid of doctors and use prayer to treat illness? Well, some researchers designed an experiment to test the hypothesis that prayer improves healthcare outcomes. Specifically, they found 1,800 heart surgery patients and followed them up over the course of a decade. And they randomly selected some of these patients to have people pray for them. And they told some of the people that people were praying for them. And they didn't tell other people that people were praying for them. And people who received prayers offered by strangers without their knowledge, it had no effect whatsoever on their recovery. And patients who knew that they were being prayed for actually had higher incidence of complications. Maybe they were stressed out, or who knows what was going on. One way or another, this demonstrates why anecdotal evidence is unreliable. The human brain does a really good job at recognizing patterns, even patterns that don't necessarily exist, so you shouldn't trust anecdotal evidence. The better way to draw inferences from data is to systematically compare the two groups using an experiment. And I'll spend the rest of today's discussion explaining the optimal way to conduct an experiment. Before we do that, let's cover some terminology. An experiment, the term gets used somewhat loosely for almost any sort of scientific enterprise. Strictly speaking, an experiment means you actively manipulate the experimental condition. That you give half of your rats a drug, the other half a placebo, that you actively intervene. Whereas an observational study is, rather than actively intervening, you just observe data that's out there. And an example of this is if you want to see if smoking causes lung cancer, for instance, it would be considered unethical to tell some people to smoke and others not to smoke and follow them up for 20 years to see who developed lung cancer. So you have to rely on observational data. Experiments are almost always more reliable than observational data, but many times you have to use observational data because experiments would be either excessively costly or unethical. So some other examples, or some other terminology related to experiments is the response or an outcome variable is the outcome of interest. It could be whether or not a person develops lung cancer. An example we're going to give here in a second, whether or not a person develops polio. An explanatory or a predictor variable is a variable that can be used to explain or predict the outcome. For example, you might think that smoking could cause lung cancer. You might think that the Salk polio vaccine will decrease your chances of getting polio. An extraneous or confounding variable is a variable that may affect the results but are not of direct interest. For examples, we'll explain here in a minute. Socioeconomic status of a child is a confounding variable in the polio vaccine, that children from wealthier families are actually more likely to contract polio than children from poorer families. If these wealthier families are more likely to get the vaccine, then that could create bias against the vaccine in this type of trial. Or for smoking, for a long time, the tobacco companies claimed that maybe there's a gene that predisposes a person both to smoke and to cause lung cancer, but it isn't the smoking that causes the lung cancer. I mean, yeah, that's obviously far-fetched, but it's a possibility that's actually very difficult to conclusively rule out, which is one of the reasons that the tobacco companies denied that their product caused lung cancer for so long. And observational units or experimental units or subjects are the persons or things being studied, the people in our observational study about lung cancer or in our experiment about a polio vaccine. So, give an in-depth example of proper experimental design with the Salk polio vaccine. The, the first polio epidemic hit the U.S. in 1916, and in the early part of the 20th century, it was a major cause of death, the majority of the victims being children. And in the 1950s, Jonas Salk developed a vaccine, and we wanted to see, is this vaccine effective? So how would you design an experiment to see if this particular vaccine is effective? Well, the first thing that you need to do in virtually any type of experiment, you need controls. One might think, okay, well, there's children dying of polio, tens of thousands per year. Shouldn't we be giving this vaccine to everyone to reduce the number of deaths due to polio? Well, that's problematic because maybe the vaccine doesn't do anything. Heck, maybe the vaccine even makes things worse, that makes you more likely to die of polio or kills you as a result of side effects. There's no way we can tell this without testing, which is why any type of drug or any type of science in general, you need to do an experiment, just test to see if it'll actually work. Next question is, can't we just compare the polio death rate from year to year? Last year the kids didn't get the vaccine. We'll give the vaccine to everyone this year and see if the death rate goes down. Well, that doesn't work well either because polio is an epidemic disease. In 1952, there were 60,000 deaths. In 1953, there were only 30,000 deaths. If you had administered the vaccine in 1953, even if it were completely useless or even harmful, you might conclude that it was working just because of the year-to-year variation in the death rate. In this case, the epidemic nature of the disease would be a confounding variable, so that isn't the proper design here. A little bit more terminology in general when you're talking about an experiment. Those who receive the drug or other form of treatment are referred to as the treatment group, and those who are not treated that are compared to the treatment group are known as the control group. So, anyway, we've established that we need to choose controls. How should we choose controls? You might think, well, in order to get treatment, you have to get parental consent to make sure that as many kids get the treatment as possible. Let's just treat the kids whose parents give consent and then use the kids whose parents don't give consent as controls. And that will maximize the number of children who receive the vaccine. The problem with that is that will potentially confound the results. They found that higher income parents are more likely to give consent, and somewhat surprisingly, children of higher income parents are also at higher risk of developing polio. Basically, children from low income families often live in dirtier places when they're growing up, so they develop mild cases of polio and consequently develop immunity. So this would create bias against the vaccine. And I mean, as it turns out, this experimental design isn't completely terrible. In fact, the National Foundation for Infantile Paralysis ended up using a variation of this design for the study that they conducted on this. Their design was the following: that you vaccinate all second grade children whose parents give consent, and you leave first grade and third grade unvaccinated as controls. As I noted earlier, this can lead to bias that you have too many children of high-income parents in the treatment group, and it potentially introduces other confounding variables as well. Polio is a contagious disease that's spread through contact, so it's possible that there's more kids who acquire polio in second grade than there is in first or third grade, which would create bias against the vaccine. Maybe there's less kids who get polio in second grade, which creates bias in favor of the vaccine. Now, if you do a large-scale study across a large number of elementary schools, this is unlikely to drastically bias the results, but it is a potential confounding variable that's introduced that we don't really need to introduce in this particular case. Oh, the optimal design, the fully randomized controlled design, is described here, that we choose controls from exactly the same population as the treatment group, the children whose parents gave consent. If your parents didn't give consent, you are not entered into the study at all in either the treatment group or the control group. And for each child whose parent gave consent, you flip a coin to assign them to the treatment group or the control group. In practice, you'd probably use a computer or something like that, but the idea is you make the assignment completely at random. The reason that you do this is you're trying to ensure that the only possible difference between the treatment and the control group is the treatment itself, that there should be no systematic variation between the treatment and the control group, and hence very little possibility for confounding. You could still get unlucky and end up with a lot of grade 3 kids in the treatment group, for example, if your sample size is small. But if you have tens of thousands of kids in the study, then the two groups should be essentially indistinguishable other than the vaccine, meaning that if the death rate is lower in one group than the other, it should be the result of the vaccine. Next important principle of experimental design is the use of placebos. You might think, okay, we'll give shots to the treatment group and we won't do anything at all to the control group, and we'll compare the two. Well, as it turns out, that's a bad design as well, because one of the really fascinating things about human nature is that sometimes patients will respond to sugar pills or saltwater injections or other phony treatments in something that's known as the placebo effect. And this slide I pulled from Wikipedia, so take it for what it's worth, but it looks like they provided references for this, and this is a list of conditions that have been known to respond to placebo treatment. And I mean, some of these are really strange, like you can apparently treat asthma with placebo, or you can treat erectile dysfunction with placebo, or pain, or all sorts of things like that. In fact, there are even cases of cancer going into remission once you inject the person with salt water. For some reason, the human brain, when it thinks it's being treated, it starts to get better, even if the treatment is having no effect whatsoever. And there's been some controversy about this with respect to antidepressants in particular recently. There's some indication showing that antidepressants do work, but there's no indication they work any better than a placebo, for example. Oh, with if so if we don't include a placebo in this vaccine trial, then it's possible that we'd see a lower rate of polio in the a lower rate of polio in the treatment group just due to the placebo effect, so we need to give a placebo to the control group. So the kids in the treatment group get a live vaccine, the kids in the control group get a placebo as the optimal study design. Another important principle of experimental design is that whenever possible, studies should be double-blind. In particular, in a case like polio, the diagnosis of polio is somewhat subjective, and if examiners know which children receive the vaccine, they might be less likely to diagnose polio among children that receive the live vaccine, producing bias in favor of the disease. So to avoid this possibility, under the optimal study design, neither the subject nor the examiner should know if a given subject received the placebo. And this type of design is called double-blind. So in other words, neither the children nor the parents nor the people examining the children for polio know whether the children received the live vaccine. That minimizes the possibility for bias. So, the results of this experiment, and it looks like part of my slide got cut off when I converted this to PowerPoint. I apologize for that. But what that's supposed to say is polio cases per 100,000. And under the randomized controlled double-blind design, we see that in the treatment group there were 28 cases of polio per 100,000 versus 71 in the control group. Interestingly, in the group whose parents didn't give consent, it was 46 per 100,000. So just between the consent and no consent group, there is a dramatic difference illustrating the importance of selecting the treatment in the control group from the same population. Under the NFIP design, we still got the correct result that there is lower polio among those who receive the treatment, but there's still the same type of bias that those in second grade with no consent get 44 cases of polio per 100,000, and in grades 1 and 3 there's 54 per 100,000, so there was significant bias against the vaccine generated as a result of the suboptimal NFIP design. Well, now I'll go through a couple other examples of experiment and proper experimental design to give you some additional insight on how a study should be conducted. So, here's an example of clofibrate, which is a drug designed to lower cholesterol and reduce the risk of death from coronary disease. I guess it was a predecessor to Lipitor or something like that. So an experiment was designed to test the effectiveness of clofibrate. Well, in this particular trial, a fair number of patients didn't take all of their pills. See that those who took all of their pills, about 15% died of a heart attack during the 5-year period, versus those who didn't take all their pills, it was 25%. So you might say, wow, it looks like it drastically reduced the mortality rate. This clofibrate stuff is actually effective. The problem with that is that this is not randomized. It wasn't like we randomly assigned half the people to take their pills and half the people not to take their pills. The people who took their pills are probably more likely to be health conscious, and they're also more likely to exercise, reduce high-fat foods from their diets, and things like that. And if you compare those who took clofibrate to those who took a placebo, we see essentially no difference in the 5-year mortality rate between the clofibrate group and the placebo group, illustrating why it's so important to use randomization when you assign experimental units to one group or the other. Another example of things that can go wrong if you use suboptimal experimental design is the porticable shunt that we discussed briefly in the first lecture. As you'll recall, if In some cases of cirrhosis of the liver, a patient can start to hemorrhage and eventually bleed to death. You can try to treat this by redirecting blood through a porticable shunt. Problem is, this is a really hazardous procedure, and it's a really hazardous procedure, and they may die of the result of the procedure rather than the bleeding. So the question becomes, is it worth the risk? Well, we saw in the first lecture that if they didn't include controls in the procedure, then it tended to overstate the efficacy of the procedure. But what I didn't show you in the first lecture was that there were 15 other studies conducted that included controls but were not randomized, that the doctor just used their judgment about whether or not this patient needed the shunt. So it may be a case where they gave the shunt to healthier patients who they thought would be more likely to survive the procedure, and hence those patients survived. Said, "Oh, this porticable shunt is great." See, it wasn't quite, the enthusiasm wasn't quite as great as when there were no controls at all, but still, the majority of the case of the studies where there was no randomization thought that the shunt did pretty well. It was only in the randomized controlled design where people were randomly assigned to get the shunt or to not get the shunt where it was more apparent that the shunt was fairly risky and perhaps not quite worth the risk. So another example that might be more relevant in the bench science world, let's say that you're doing an experiment on laboratory mice. You grab 6 mice, put them in the treatment group, take the 6 mice that you don't grab and put them in the control group. What's the problem with this approach? Well, there's a couple problems with it. One is that it's not random. What if the 6 mice that you grab are older and slower, less healthy, whatever the case may be? That could bias and confound the effects of your experiment. It's also not double blind, that the researcher knows which mice are the controls. The optimal design, which I didn't put on a slide, would be to just give a number to each mouse, use some sort of random number generator to generate the 6 mice, and in the ideal world, you have the person recording the data does not know which is in the treatment group and which is in the control group. Now, obviously, many times the person who is performing the injections will be the same person who's recording the data, in which case making it completely double-blind is impossible, but if it's possible to make it double-blind, it should be double-blind. Another bench science example is, let's say you're doing a microarray experiment, and say you want to compare the effect of 2 treatments on a cell line using microarrays, and samples with treatment 1 are labeled with red dye, and samples with treatment 2 are labeled with green dye. And so what would be the problem with this approach? Well, the problem here is that there may be dye bias going on in the experiment. Maybe green dye just always shows a slightly higher expression level than red dye. If that's the case, then as you see in this graph here, it could end up biasing your results. A better design would be, and we'll discuss this in more detail in a future lecture, but a better design would be to give green dye to half of the treatment group and red dye to half of the treatment group. Likewise, give green dye to half the controls, red dye to half the controls, to try to control for this possibility of dye bias. This is called blocking, and I'll discuss it in more detail later. Another important point is that randomized controlled experiments are not always possible. They may be too expensive, they may be unethical to do a randomized controlled treatment. For example, well, I mean, this is kind of a ridiculous example, but it was in the Onion and I thought it was kind of funny. Say, like, heart transplant group significantly healthier than heart transplant placebo group. If you do placebo heart surgery on someone, then they die. So the people who get true heart transplant treatment are more likely to survive? Well, aside from the fact that experiment is unethical, it's also ridiculous. You could never actually do that. So basically, at the point where placebo heart surgery is unethical, all you can do is some type of observational trial in this case. As I said, a randomized controlled double-blind experiment is the gold standard of experimental design. Anytime you can do this design, you should do this design. But in many cases, randomized controlled experiment is going to be impractical, super expensive, or otherwise unethical. Aside from heart transplants, as I said before, smoking is a good example of some place where you have to use observational data, that you can't just assign half of your study to smoke and another half to not smoke and follow them for 40 to 50 years to see who gets cancer. Aside from the fact that would be ridiculously expensive, it's also highly unethical. So many times you just have to design an experiment that's as close as possible to the gold standard and acknowledge the limitations. And this is an area where people sometimes struggled in some of the first homework assignment where you say, well, this is suboptimal, they should have done it this way instead, and the way they suggest to do it instead would have cost like $10 billion. And well, yes, that would have been a better design, but the government is probably not going to give you $10 billion to run your experiment, so you just have to make do the best you can with the best data that you have. So, things to remember for today is that experiments always need to have some type of controls, and you want to make the controls as similar as possible to the experimental units receiving the treatment. And if the treatment involves giving a drug or a treatment to a live subject, you always want to use a placebo. And subjects should be, whenever possible, subjects should be designed to the treatment completely at random. If you use other criteria, you can introduce bias. And if there's potential confounding variables, such as the dye example and the microarrays, you should use randomization to offset that. We'll discuss that in more detail later. And whenever possible, experiments should also be double-blind.